Welcome back to Chapter 21, Part 3. Here we're going to really explore the conditions of the Hardy-Weinberg equilibrium and really look at the causes of evolution. So the first thing that we're going to do is understand that while the Hardy-Weinberg equation does describe a hypothetical population that is not evolving, right? Real populations evolve. I mean, real populations absolutely have allele frequency changes and genotype frequency changes over generations. So what are the things that cause these changes? There are 5 basic things that cause the changes. And the first time we go through these, we're going to go through them like Hardy and Weinberg did, where we're going to think about them as ways that populations don't change, right? Again, that is how Hardy and Weinberg actually figured out their math. So we're going to go through these as ways that evolution does not occur, and then we'll talk about how if we violate any one of these conditions, then evolution will occur. So the first one is no mutation. No mutations. If you have no mutation, that means that the gene pool is not going to be modified at all. If that condition doesn't hold, if we do have mutations, then the gene pool is going to be modified. And if mutations occur, it is possible that, entire genes could be removed, entire genes could be duplicated, right? We went through all of that in part 1. So the condition for no evolution is no mutation. If we violate that condition, then we're going to end up having some change in allele frequency. The next one is random mating. So random mating is you're not assorting, you're not choosing your mate, just mating with whoever's nearby. I like this little cartoon here. What happens if you violate that condition? If individuals mate within a subset of a population, such as like neighbors or close relatives. By the way, mating with close relatives can be fairly common in the world, in the animal kingdom and plant kingdom. We call that inbreeding. And so just heads up, I know we have like a thing about inbreeding in humans, and rightly so, but it's not like it's uncommon amongst many organisms in the world. So random mixing of gametes doesn't always occur. In fact, it doesn't happen all that often. And so that can cause genotype frequencies to change, and which can lead to allele frequency change. And of course, that is evolution. So for no evolution, we have random mating. For evolution, we have something we call assortative mating. We would also have to have no natural selection for no evolution to occur. If you violate that, if you have allele frequencies change when individuals change or differ in their amount of survival or reproduction, then you have natural selection. If you want no evolution, you have to have no natural selection. It helps to have a really large population size. If you happen to violate this one, you would have a small population size. Then here's the thing about small populations. Small populations have allele frequencies that fluctuate just by chance. And we'll talk about genetic drift as that chance thing that happens. But if you have a really large population, then your chances of it just shifting due to chance alone are very small. You can think about it like this. If you have a small bag of M&Ms, right? And you pick out one M&M color, you pick out the blue one, that actually changes the frequency of the blue M&Ms in that bag. But if you have like 10 family-sized bags of M&Ms and you pick out one blue one, it really doesn't change the frequency that much. And so the population size being large, you're less likely to have evolution. The last one is gene flow. And gene flow is when organisms move from one place to another. We often use the word migration, and this little image right here shows migration, but gene flow is the proper term. As long as you have no movement from one population to another, you might not have evolution, and it's absolutely required for no evolution to occur. When you move alleles from one population to another, that can change the allele frequencies of both of the populations, of the population where you're removing it from and the population that it's moving to. So we're going to look at an example of how all of those factors might work in a gene for phenylketonuria, and this is in humans. Now, the gene for phenylketonuria is in Hardy-Weinberg equilibrium. And I don't know if you remember anything about phenylketonuria from before when we were doing genetics, but phenylketonuria is a gene where if you have it, you have this disease called PKU. And PKU is potentially quite deadly disease. The problem with PKU is that if you have the disorder, then you are unable to take the amino acid phenylalanine and turn it into tyrosine. And there's the word tyrosine right there. Both of those are amino acids. And this is a healthy thing to be able to do. It's the only way that we can make tyrosine in our bodies is if we have the right enzymes to change phenylalanine into tyrosine. But in PKU, you don't have the right enzymes, and so you don't make the tyrosine, and unfortunately the phenylalanine starts to build up in your tissues and in your blood and in your brain. And this can lead to very, very difficult consequences, including the deterioration of brain function. But for PKU, we are in Hardy-Weinberg equilibrium. What does that mean? It means that there's not a lot of mutation that's happening at that PKU gene site. So mutation rate is low for PKU, as it is low in general for most genes in eukaryotic organisms. Mate selection is random with respect to PKU. And what that means is that if you are heterozygous, you probably don't know it. And so you're not like choosing your mate based on whether they're heterozygous or not. So that it's just kind of random whether or not the person you are having babies with has that PKU gene. Now, natural selection does act on PKU, but because it's so rare in the population, it doesn't really end up changing the frequency of individuals who have PKU. And these days, one of the first things that happens when an infant is born is that they are tested for whether or not they have PKU. It's like they do this little heel prick, and they test to look for PKU. If the baby has PKU, then they have to go on a special diet. But these days, going on that special diet is fairly easy to do. So natural selection is only going to act on individuals that don't know they have PKU. And if they do know it, maybe they're not following the dietary restrictions. The next one is that the population is large. Yeah, the population is large in the United States. And so we have another reason why that PKU is not evolving in the United States. And what we see is that the population outside the United States has a PKU allele frequency very similar to populations of those in the United States. And so we have evidence that even if there was gene flow, that it wouldn't be changing our allele frequency in the United States. So PKU, not evolving. If we look at the math of this, we can say that, remember, we don't really know how many people in the United States are heterozygous per se. Like you probably don't know if you're heterozygous for PKU or not. I actually know because I had some genetic testing done. So I'm not heterozygous for PKU. But we can actually calculate all of the parts of the Hardy-Weinberg equation just by knowing how frequent the homozygous recessive individuals are. Remember that Q squared is defined as the number of, or the frequency of the homozygous recessive. So I'm going to remind you of that. I'm going to have this over here. The frequency of the homozygous recessive. So I'm going to do like little r in this case, because like r is easy to draw. So there you go, frequency of the homozygous recessive. And remember, that's Q squared. So if we know that PKU occurs in 1 in every 10,000 births, then we can calculate Q squared super easy, because all we have to do is a frequency. 1 divided by 10,000 is equal to 0.0001. Now we can calculate the frequency of the alleles, not because, the other way we did it was we counted up how many heterozygotes there were, we multiplied times 1, and right, we did that elaborate math there, but we can kind of estimate the frequency of Q by just taking the square root of a square. So this is Q squared. If we take the square root of it, then we get 0.01, and that is Q. Remember that we can calculate P by just doing 1 minus Q. So 1 minus 0.01 is equal to 0.99, and that's what P is equal to. We can calculate P squared if we want, but let's do, since we're interested in whether or not this trait can be passed on, let's do 2PQ. Only heterozygotes can have babies that are having the PKU disorder. So 2 times P times Q. Remember that P was 0.99 and Q is 0.01. So 2 times 0.99 times 0.01 gives us 0.0198, which is about 2%. And it turns out that about 2% of the US population is heterozygous. So this is, since it seems to remain at about 2%, we know that evolution is not happening. Now, there are 3 major factors that alter allele frequencies, and there are some minor ones too, but we're going to really focus on the major ones. And remember, the Hardy-Weinberg assumptions were no natural selection, no mutation, no gene flow, have to have a large population, and have to have random mating. Of those 5, the 3 that are probably the most important ones that have the biggest effect are natural selection, and then genetic drift, and then gene flow. So over here, we would be violating natural selection. We would be violating this migration one, which is officially and better called gene flow. And then we would be violating this large population one. So we'd have a small population. And genetic drift is associated with small populations, which we'll learn about in a little bit. But let's start out here first with natural selection. If you violate the no natural selection, that means you do have natural selection. And we learned previously that differential reproductive success, passing on certain alleles more in greater proportions than other alleles due to environment, differences in environment, that's natural selection. So an example for this might be the use of DDT, which is a pesticide. DDT was sprayed extensively in the 1960s and 1970s. It's no longer in use, but what happened in some of these fruit flies, and this is what D. melanogaster is. D. melanogaster is Drosophila melanogaster, and that's a very common fruit fly. Widespread, found in agricultural areas, and what happened during the time of DDT spraying is that these fruit flies evolved to be resistant to that pesticide. So here's the first generation where before the application of the pesticide, here's the pesticide right there, and it killed a bunch of the animals, a bunch of the fruit flies. But some of them, just by chance, happened to have a mutation that allowed them to survive that. Others, maybe they weren't actually, maybe they weren't near enough or didn't get hit by the pesticide, so they survived too. So in a later generation, you've got more of the pesticide-resistant fruit flies and less of the ones that are resistant, or sorry, are susceptible to the pesticide. After another round of spraying, you'd have most of your fruit flies not able to be killed by the pesticide, and only a few would still be able to be killed by that pesticide. Now, that is natural selection. We'll talk a little bit about natural selection versus artificial selection. I think we already talked about that actually. But we put that under the category of natural selection. Those fruit flies that are resistant are the ones that are adapted to this new environment of putting down the pesticide. So natural selection causes adaptive evolution. And again, that's just a process in which traits that enhance survival, right? We're going to have some enhancing survival or reproduction. They're increasing in frequency over time. What's genetic drift? Genetic drift happens in small populations. And the reason that the populations change is due to chance alone. I gave you an example of M&Ms earlier. That's kind of what genetic drift is like. It's a population which is small, and allele frequencies fluctuate unpredictably. You literally cannot predict what's going to happen in the next generation because of just these chance events. So in this population right here, we've got a distribution of red, pink, and white flowers. And by chance and chance alone, only a few of those reproduce. And it turned out that the ones that are reproducing, the red ones didn't happen to reproduce for whatever reason. Maybe somebody picked the red flowers. They picked more red flowers. So flowers are the reproductive organs. And so, right, you get this chance event where only these ones are reproducing. If that's the case, then the population will have changed over time, right, from one generation to the next. And so the ones that survived reproduce, and we get more white and pink flowers in this generation. Now, in the next generation, maybe by chance events, only the red ones got a chance to reproduce. And so in that next generation, we have a population that is fixed in their alleles. There's no more variation. Again, in a small population, chance events like this definitely happen. And we've had fluctuation in an unpredictable manner from one generation to the next, where we started out with P is 0.7 and Q is 0.3. Then it went to 50-50. And then it went from P is fixed at 1 and Q is at 0. Remember, P plus Q equals 1. So that makes sense. Now, we have a couple of different ways that genetic drift can happen. There's the first one is called the founder effect. And the founder effect happens when a few individuals become isolated from a larger population. Often we kind of think about this as they get stuck on an island or something like that. So they found, that's actually the way you say that, is these individuals found a new population. And maybe that's on an island. Maybe they got blown off course or something along those lines. This small population of founders does not represent the allele frequency of the original population. And so just by moving from one place to another and being a small subset, your descendants do not represent the same allele frequency as the original population. That's evolution, just by moving or founding this new thing. Now, this is not considered migration. And that's why we don't use the word migration very much when we talk about gene flow. So I'll explain that more when we get into the subject of gene flow, just so you know. But for now, just remember that the founder effect is an example of genetic drift and not gene flow. The other example of genetic drift is the bottleneck effect. And the bottleneck effect is very interesting because what happens is the whole population has a drastic reduction in population size. And this is usually due to a sudden environmental change. So, so the gene pool may not represent the original population because it's just such a dramatic decrease in the population size. If the population size remains small after this, you can actually get more genetic drift happening. So what does that look like? Think about this is the original population. This is all the diversity. We've got blue and white and yellow in here. And then over time, we have the bottlenecking effect. So only some of them, if you pour this bottle of gumballs or whatever they are out into a jar, only a few of them are going to make it into that jar, right? Because they have to go through this bottleneck. And the surviving population does not look and does not have the same allele frequencies as the original population. We see evidence of bottleneck effects in lots of organisms that have gone through reduction in genetic variation. For example, this is a Florida panther, and it is a subspecies of the American panther, and it is in danger of extinction because humans hunted this organism into near extinction. So it went through a bottleneck. Remember that bottleneck effects are chance events. So even though I was talking about hunting of the puma, of the panther, humans weren't picking which puma to kill. We were just trying to kill them all. So, but a few did survive, and which ones survived was due to chance alone. So we have this panther with very little genetic variation, and it is in danger of becoming extinct. Another example of this bottleneck effect or in general genetic drift, is the greater prairie chicken. I like this example from your book because it's about Illinois. So, and we live in Illinois, or most of us do, at least in my class. So, what caused this bottleneck effect was the loss of the prairie habitat due to increased farming. So reducing prairie habitat put a reduction in population of this prairie chicken that lived in this habitat. And this is a native of America. So this prairie chicken, which looks a lot like a domestic chicken, had lived in America prairies for, millions and millions of years. So with all of this conversion of prairie to farmland, in Illinois, fewer than 50 birds survived, and this drastic reduction in population reduced the genetic variation. And the result of this was that only 50% of eggs were hatching because they were doing a lot of inbreeding, which we mentioned earlier. Too much inbreeding can lead to reduction in fertility. So 50% of their eggs hatching, this population is in deep trouble. And again, the cause of that was loss of habitat, which caused randomly some to be in prairies and some of them were in farmlands. And so it ended up increasing the frequency of harmful alleles due to inbreeding. So this is just a map of where the prairie chickens were found before the bottleneck, and then farming happened, and these were like two tiny little populations in 1993 with where there were some prairies existing and just those two locations. So a huge drop in environment caused a drop in the population size. So again, we can look at just some raw data here. In Illinois in the 1930s to 1960s, there was a census taken of those prairie chickens. There were 1,000 to 25,000 If you go on to Bio 102, you can learn about how populations are estimated. The number of alleles per locus was 5.2. Remember that we talked about heterozygosity earlier in this chapter. Heterozygosity is the number of genes that have different alleles at their locus. So a heterozygosity number, and the percentage of eggs that hatched were 93%. By 1993, the population size was less than 50, heterozygosity numbers had decreased, and the number of eggs hatching had dramatically decreased. If we looked at those same species in Kansas and Nebraska, where there's no bottleneck, we see that the percentage of eggs hatching was still quite high, and so those are healthy populations. So the scientists did what researchers do when they're trying to figure out things, and what they did was they added 271 birds from a population from Kansas and Nebraska to the Illinois population. And when they did that, they increased the heterozygosity, they increased the number of alleles in the population, and the egg hatching rate improved to over 90%. So by doing this, basically what happened was that we saved the population of birds in Illinois. So just to summarize, genetic drift happens in small populations, and it's a random change in alleles. There's no specific cause for the reason why one allele does better than another, unlike with natural selection. Genetic drift can lead to a loss of genetic variation, a dramatic loss in genetic variation in that population. And it's also possible for harmful alleles to become fixed, especially in that small population if inbreeding is happening. Okay, so we're going to talk about gene flow now. And remember that we sometimes use the word migration in place of gene flow, but that's a really bad idea. It's helpful to just kind of as a mnemonic to remember what gene flow is. But migration and gene flow are really different things. Gene flow consists of movement of alleles from one population to another. So, and this is like a permanent thing. So we're moving from one population to another. Before, when I was mentioning the founder effect, I said that was not the same thing as gene flow. So I just want to remind you of that slide and how it's different. With the founder effect, a subset left the population but didn't join another population. It just left that population. With gene flow, here we are back with gene flow. With gene flow, you're leaving one population and you're joining another population. Okay? So with gene flow, alleles can be transferred through either the movement of individuals or it can be the movement of gametes. Now, in humans, we don't think of our gametes as moving separate from us, but with plants, that does happen. Pollen can blow around a lot, and so that pollen can go from one population to another by itself. Gene flow, unlike genetic drift, gene flow tends to reduce variation among populations. So if this is the original population and this is the neighboring population, and this one has a lot of variation and this one doesn't, by moving some of these alleles into the original population, you change the original population. You make it more diverse, more variation, and you're not really affecting too much the population that left, the population where the people left or the individuals left, because it hasn't changed its allele frequency very much. But they're becoming more similar to each other, right? Through gene flow, the genetic makeup of those two populations becomes more similar. Sometimes gene flow can be detrimental to organisms. And the example that we're going to go through is the Lake Erie water snake. So I know some of you don't like snakes, but they're very interesting. Take a look at these snakes. Some of them are solid in color, and light or a little bit darker. Some of them have spots. Some of those spots are very light spots. Some of those spots are very dark spots. So lots of genetic variation in these Lake Erie water snakes. What we've found is that a strong banding pattern with the spots or the stripes, almost like stripes right here, is favored on the mainland. And the unbanded snakes, like this one and this one, are favored on the islands because it allows them to camouflage a little bit better on the islands of Lake Erie. So migration of banded snakes from the mainland population to the islands actually disadvantages the snakes on those islands because they start to have more banding on the islands. And really, it's better for those island snakes to have less banding. Take a look at this map-wise thing. So here's Lake Erie, right? And there's all these islands in Lake Erie. Here's Canada, here's the United States, and the unbanded versus the banded. So on the mainland of Ohio, we have a lot of banded, that's the type A, and type D is unbanded. And on the islands in between, there's way more unbanded. And then if we look at Ontario, like basically all they have are the banded snakes. So that's, of course, these populations from Ohio and Ontario keep feeding into the islands as a form of gene flow. Gene flow can actually improve adaptation. So we were just talking before about how gene flow can reduce adaptation, but it can improve adaptation to local conditions. For example, there are alleles for pesticide resistance in a mosquito called Culex pipiens. And I'm using the same diagram that I used for when we were discussing the insecticide resistance for the fruit flies. It's the same concept. So, but this time we're talking about a mosquito, and this mosquito is a vector or carries the West Nile virus. So again, you're going to have to think about this in terms of advantage to the mosquito. And I know that's hard because West Nile virus is dangerous to humans. But again, just keep in mind that the mosquitoes are doing okay. And that's the example that we're stepping through. So imagine that we've been spraying, and we do this in my town where I live. They spray to kill mosquitoes. They spray all summer long, like about once a month in my neighborhood. And of course, some of the things that they're killing are the mosquitoes that carry West Nile virus, but some of those mosquitoes that carry West Nile virus are resistant to the insecticide, the pesticide. And so those are the ones that are increasing in the population. And you can spray and spray and spray all you want, but if the population is mostly resistant, then you're not going to be killing many of those individuals. Now, my area sprays for mosquitoes, but maybe your town doesn't spray for mosquitoes. So where gene flow becomes an improved adaptation is where if my mosquitoes move into your area, my mosquitoes that have been have evolved resistance, and they move into these other areas, and they're like, oh, we got all these mosquitoes and they're carrying West Nile virus, let's spray for them. Well, they're already kind of pre-adapted for surviving in those areas because they already have developed that insecticide resistance, that pesticide resistance. So gene flow can improve. And again, for us, that's not such a good story, right? But for the mosquitoes, that gene flow has improved adaptation to local conditions. Another interesting thing about this is what's going on with humans. And we, modern humans, are able to travel way more than we've ever been able to do in the past because of modern technology, like the airplane. So what that means is that gene flow has become very important in the evolution of modern human populations. again, gene flow has changed modern human populations quite a bit. So just to kind of wrap things up, natural selection is, of the ones that I've mentioned so far, and of any of the 5 that we will be continuing to talk about, natural selection is the only mechanism that consistently causes adaptive evolution. Evolution by natural selection involves some chance, because remember that mutation is a chance event, right? And mutation causes variation. So there is some chance involved, but there's also sorting. And what I mean by sorting is that beneficial alleles are favored by natural selection, so they're allowed to go into the next generation. And that's a sorting kind of process, or a non-chance process. So when a lot of people think that natural selection is just chance, they're really missing out on the main process. Yeah, it might be chance that you have a particular mutation or not, but whether or not you survive based on that mutation is not chance. That's sorting. And natural selection does increase the frequency of alleles that improve reproductive and survival advantage. All right, that was a long one. We covered a lot of ground in that section. We're moving on to chapter 21, part 4. I'll see you in the next video.